Hello everyone, welcome you all in the another video tutorial. So in this video lecture we will discuss about X-ray crystallography. So let's start with the introductive part. So basically the X-ray crystallography is an experimental science method, or it is a scientific method which is used to determine the arrangement of atoms. So basically this method is used for determining the atomic and molecular structure of any crystal, in which the crystalline structure causes a beam of incident X-rays to diffract into many specific directions. Now this technique uses X-ray light, which is having wavelength of minimum 1 angstrom or 10 to the power minus 8 centimeter in length. So we know that what are the X-rays. So the X-rays are also called as X-radiation, which are a type of high energy electromagnetic radiation, and most of the X-rays have wavelength ranging from 10 picometers to 10 nanometers. So due to their specific wavelength, these X-rays are used to identify the structure of the crystals of different types of molecules. So in the present scenario, the X-ray crystallography is most favored technique to determine the structure of different proteins and biological membranes in the field of biology or in molecular biology. Besides this, the method of X-ray crystallography is used in different types of mechanisms to determine different types of structures of different metals or other crystal forms. Now let's understand some characteristic features about the X-ray crystallography. So it is a technique which is used for determining the atomic or molecular structure of a particular crystal. Or for using the X-ray crystallography, the matter should be in the form of crystal. So basically it is crystallized by using different methods. So in this technique, the crystalline atoms causes a beam of X-rays to diffract them into many specific directions, and then the structure of these crystal is determined by the use of these X-rays. So basically in this method they use an X-ray beam and hit this beam to the crystallized molecule of the particular molecule whom structure is to be determined. Now the electrons surrounding the molecule diffract as the X-ray hit them, and this diffraction form a pattern, and this type of pattern is called X-ray diffraction pattern. So it is also called that in the form or in the method of X-ray crystallography, the X-ray diffraction method is used. So let's start with the history and discovery about the X-ray crystallography and X-ray diffraction method. So before the X-ray diffraction method, a scientist named as Wilhelm Röntgen discovered the X-rays in 1895. So before this, physicists of all the world were uncertain of the nature of X-rays, but after that they suspected that the X-rays were also a type or a waves of electromagnetic radiation, and they are also a form of light. But sooner it is also suspected when a scientist named as Sir William Henry Bragg determined about the crystal structure by using X-ray diffraction method in 1907, and he also proved about the unpopular and the observation of X-ray diffraction used by Max von Laue in 1912, and he confirmed for most of the workers and researchers from all over the world that X-rays are a form of electromagnetic radiation. After that, using the X-ray data, Watson and Crick were able to determine the helix structure of DNA in 1953. So Watson and Crick were also awarded by Nobel Prize for their discovery and to discover about the alpha helical structure or double helical structure of DNA in 1953. So before moving to the procedure of X-ray crystallography, let's understand first about the X-ray diffraction and about the Bragg's law. So basically, let's understand first that what is the meaning of the word diffraction. So the diffraction is defined as the slight bending of a light when this light passes through the edge of any object. It bended, and this bending is considered as diffraction. So the X-ray diffraction is technique where the X-rays are used due to their wavelength, and this wavelength is similar to the intercellular spacing between the atoms of the sample, and that's why specifically X-rays are used in the method of X-ray crystallography. So this method of X-ray diffraction is used to determine the sample's composition or also its crystalline structure. So here the electrons which are surrounding the molecule diffracts as the X-rays hit them. So the beam of the X-ray light is used to hit the molecule, and this hitting form a pattern, and this type of pattern is called as X-ray diffraction pattern, as this method is called as X-ray diffraction. So the angle of this diffraction is determined, and to determine this method it can be used the differences between the atomic planes which are used by Bragg's law. So Henry Bragg also suggested a law to identify and to measure the angle of diffraction in the method of X-ray diffraction. So Henry Bragg suggested that there is a definite relationship between the angles at which the beam of X-ray fall on the parallel planes of the atom in a crystal, of which structure is going to be determined, and in that order there will be strong reflection that the wavelength of the X-rays and the distance between the crystal planes will be 2d sin theta equal to n lambda. So here Bragg also suggested an equation to determine the angle of diffraction, and this equation is sin theta equal to n lambda upon 2d. Here the d is the space or intercellular space between the diffracting planes, and the theta is incident angles, and in is an integer, and the lambda is defined as the wavelength of the beam of X-ray. So this is the Bragg's law which is used for the X-ray diffraction to find the angle of diffraction of these molecules. Now let's understand about the procedure of X-ray crystallography. So the process starts with obtaining an adequate crystal of the material, and this is the most difficult and most important step of the whole procedure, because it's very important to found a crystal or crystallized material, and it is the most difficult method to make a crystallized form of any material. Now the second step includes where the crystal is placed in an entrance beam of X-rays, and these X-rays are hit in these molecules. After then, the crystal is rotated to the different angles so that it can record every orientation of the crystal, and after that the regular pattern of reflections can be made. So for this method only the monochromatic X-rays, which usually have a single wavelength, are used. Now after that the data which is found by this orientation or by this procedure is combined with the chemical information of the sample, and multiple data sets have collected, by which each set covering slightly more than half of a full rotation of the crystal, and they contain thousands of different reflections. After then, in the final step, the procedure of model of rearrangement of atoms occur, where these data is used to analyze the crystallized structure of the particular molecule. So after the diffraction pattern is obtained, the data is then processed by the help of computers and other systems, and the structure of atom or the molecule is visualized. So this is the procedure for X-ray crystallography. So now let's discuss about the significance and applications of X-ray crystallography. So as we have discussed that the method of X-ray crystallography is used to determine the molecular structure of any crystalline material. So it is used to identify the crystalline phases and the structural properties of different types of crystallized materials, and it is also used to measure the thickness of any thin film and also of any multi-layer film. The method is also used for identification of unknown crystalline materials, different types of inorganic materials or minerals which found anywhere. It is used for the identification of these materials in the field of industrial applications. So in the industries it is widely used like in the fields of geology, environmental science, material science and many other fields. This method is used for the determination of unknown solids which are very critical to studies. Now the method of X-ray crystallography is also used in biology. In the field of molecular biology, it is used in proteomics. So now let's understand that what is the proteomics. So basically the proteomics is defined as a branch of biotechnology in which the study about proteins, their structure and their interactions is studied. So here the method of X-ray crystallography is used to study about the protein's structure, interactions of proteins and several conformational changes made by different type of structures like primary, secondary and tertiary structure of proteins, and there are different types of catalyzing methods adopted by different types of enzymes which can be determined by the method of X-ray crystallography. So X-ray crystallography also used to determine the structure of different proteins and enzymes, because we know that enzymes are also proteinous in nature. So these are some significances and importance of the crystallography method. Besides these significances and importances, there are several limitations of the X-ray crystallography also. The most concerned significance or the most concerned limitation is it is most difficult method to crystallize any material. Besides this, there are several small molecules which are very easy to crystallize as compared to the macromolecules, but the X-ray crystallography is possible even for viruses only when it is in their crystallized form. So it is difficult method to crystallize the macromolecules like several proteins and other macromolecules. So these are the limitations with this method. So in this video we have discussed about the crystallography, its procedure and its applications in various fields. Thank you.